welcome to the microbial world. Today I'd like to give a presentation about the most common event in living cells. What might that be? It has to do with bioenergetics, and it has to do with ATP, the general energy source for metabolic reactions. And we are starting to look at the bacterial cell. And here you see a simple scheme. We don't know what species of bacterium that is. It's feeding on glucose. Here it has a membrane surrounding the cell. It's consuming oxygen, and you can see glucose is taken up into the cell by means of a transport system here, and then it's used for biosynthesis, and biosynthesis requires ATP, and the ATP is made by a protein named ATP synthase or membrane-bound ATPase. Okay, and here on the left side you see to make this ATP, part of the glucose is oxidized to CO2. This generates electrons or reducing equivalents, and these are flowing through an electron transport chain or respiratory chain over several steps. We don't care about the steps at this moment. And finally, oxygen is reduced to water. And when you look what this electron transport chain does is, finally, it transports protons, hydrogen ions from inside to outside. And actually, one should think about renaming this to proton transport chain, because this is the most important thing it brings about. And these protons are coming back very rapidly via the ATP synthase, and because the protons are not produced newly but just cycling across the membranes, this processes are named bacterial processes. And okay, we looked at the bacterial cell, but you should know the same principle of proton cycling is going on in mitochondria, which actually are former bacteria, and in chloroplasts. So no matter whether this is respiratory electron transport or photosynthetic electron transport, it's the same principle that we have. Proton cycling is the most common process in living cells. And the idea and principle of this is coming from Peter Mitchell. He has got the Nobel Prize in 1978, and he has found out that bioenergetics is based on bacterial or cyclic translocation of protons across closed membrane systems. So you need a closed membrane system for that. And the transmembrane proton concentration gradient, or delta pH, and the electrical membrane potential contribute to the proton-motive force, the power that moves protons across the membrane. And I would like to answer the question, how many protons cross the membrane every second in a single bacterial cell? And I couldn't find this number in the literature or in textbooks, and therefore I thought about how to calculate that. I'm going to demonstrate it. So lots of the numbers I'm using for these calculations are coming from this very good database of useful biological numbers from the Harvard University. You find the address here. And okay, the first so-called bionumbers are also in the textbooks already. So the question is, how many protons does the ATPase or ATP synthase use to make one ATP? And the number is 3 to 4, and it might vary, but it's in that range. And we have learned already, when you make an ATP and take up proton, these have been translocated just before by the respiratory chain. So that means you have to multiply the number here with 2. And the next number is how many ATPs are conserved by the so-called oxidative phosphorylation when a glucose molecule is oxidized with O2. And this number is round about 30. We don't care about specific respiratory things. It might be in mitochondria, in bacteria. We don't care. It's around about 30. And if we bring all these numbers together, we can find out how many protons are translocated if one glucose is oxidized with O2. And here are the numbers. 3 to 4 protons per ATP times 2 is 6 to 8 protons per ATP translocated across the membrane, times 30, you end up with 180, and multiplied with 4 with 240 protons per glucose. Quite a big number. Remember, when glucose is oxidized, the release of the reducing equivalence is 24, and 10 times more are the protons that are translocated across the membrane of the respiring cell. Okay, but now I would calculate this per second in a single cell. And I have more bionumbers from this database, and I have the volume of a single cell, the dry mass, the ATP concentration in the cytoplasm, the ATP turnover for maintenance, so per dry mass per hour, and also during growth of cells. And finally, the number, how fast is the ATP synthase, so how many ATPs can it make per second. And sorry for all the numbers, just stop the video if you would like to look at them in more detail. I just show them and you can just overlook what's going on here. So the volume is about 0.5 micrometers to the third, or 0.5 times 10 to the minus 15th liter. Okay, this is just a number, very small. The dry mass of a single cell might depend on the growth state. So resting cells, stationary cells, might have 200 femtograms, and a femtogram is 10 to the minus 15 grams. Or the growing cell, these are really fat cells, they are definitely growing. They have 2 times or even 2.5 times more biomass and would also have more volume. We don't care about the details so much. So the ATP concentration in the cytoplasm is about 2 millimolar in resting and 5 times more in growing cells. And from the literature, you also get these numbers, or from this database, ATP turnover per gram dry mass and hour in millimole. And you see 8 is for resting cells and 60, so 7 to 8 times more for growing cells. And we would like to calculate this for a single cell. Therefore, we have all these numbers. And last but not least, the turnover number says an ATP synthase enzyme can produce 33 to 100 ATPs per second. And remember, per proton, for the proton, this means 3 times to 4 times more. And then you have 100 to 400 protons are channeled through the ATP synthase into the cell. Okay, sorry for all these numbers. It will be over in a minute. So we calculate now everything for a single cell. And then it says we have an ATP turnover per cell of about 250,000 in resting and 2 million in growing cells. 2 million ATPs in a single cell. 250,000 in a resting cell. The ATP concentration metabolized per second, if you calculate this for the volume of the cell, the cytoplasmic volume, is about 0.8 millimolar for resting cells. It will be more for the growing cells. However, the cells are also bigger, so I didn't add this number. It will be a little bit more complicated, at least 0.8 millimolar for resting cells. And perhaps you have remembered 2 millimolar was the concentration in the resting cell. So that means every 3 seconds you will have the complete ATP content turned over in a resting cell. The next number is very easy to calculate. If ATP synthase produces 100 ATPs per second, you will need for 250,000. This number is divided by 100, and that means 2,500 copies of the ATP synthase are required around the membrane of a E. coli cell. And last not least, and with the red frame, you see the protons translocated across the membrane per second. And this is 1.5 million, so 6 times this number, or even 8 times this number. That means 16 million protons cycling, shuttled across the membrane in growing E. coli cells. Okay, huge number. And I can conclude from this findings, a proton crossing a membrane is the most common event in bacteria, and it's the same for the mitochondria, and it's the same for chloroplasts, and this means it's actually the most common event of life. Okay. Let's learn a little bit about the proton-motive force and answer the questions. What is the power that moves the proton across the membrane? Which gradients are contributing to the PMF? And finally, why is the delta pH less important than the electrical membrane potential? And for that, we have to do a look into a cell. Again, we have already learned 1 to 8 million protons are cycling here, and we have learned there is a membrane potential, because when protons are pumped out, there will be a short come of the positive charges, and we will generate a membrane potential here. And we will look at these numbers. And okay, I forgot a quite important point. These transport systems, as actually all transport systems, are working reversibly. That means cells can perform reversed electron transport by taking up protons. They can rise the negative potential of electrons. And the ATP synthase can pump out protons and can use ATP to pump out protons as well. So these are working reversibly. This is quite important to know. Okay, and now we are going to have a look into the cytoplasm and into solute concentrations in E. coli, and everything is given in millimolar or millimole per liter, and we have quite interesting numbers there. So the most prominent cation is potassium, K+. This is about 200 millimolar, and that means you have 6 million K+ in a cell. And now comes the astonishing number. The protons are there only 6 million times less than K+. There are 6 protons in a cell. If you take a snapshot at a certain situation, you will see 6 free protons. How can that be? Okay, the pH is about 7.7. Let us say it's 8. And pH 8 means 10 to the minus 8 mole protons per liter. And the cell has a volume of only 10 to the minus 15 liter. So this number still has to be divided by 10 to the minus 15th. And this adds up to 10 to the minus 23rd. And remember, a mole was 6 times the Avogadro number, 6 times 10 to the 23rd, times 10 to the minus 23rd is just 6 protons in a cell. Seems unbelievable. But the explanation comes here. So there are many largely negatively charged solutes in the cell. And these are the amino acids. These are the nucleotides and other metabolites that are functioning as very strong buffers. So the most prominent one is glutamate. You have the other some other amino acids. Also the nucleotides with the phosphate groups, they are like phosphate buffer. And you have the carboxylic groups and amino groups, and everything contributes to the fact that the cytoplasm is very well buffered. And that means millions of protons can get in or out without heavily affecting the concentration of free protons. Okay, let's have a look at the membrane as well. And what we should know is the cytoplasmic membrane, or the inner chloroplast or mitochondrial membrane, is an electrical condenser or capacitor, and this is charged by chemical reactions. And these chemical reactions are performed at primary transport systems. It's also called primary transport. And then we have also symport or antiport of ions. This is a simple scheme of that. So this might be an ATPase that is translocating protons and making a chemical reaction, which is not indicated here. And this might be a respiratory chain, which is pumping out protons and having some redox reactions going on. And you could reverse it because it's reversible. And then there are other things, secondary transport systems that take a proton to transport something else, or there's a symport or an antiport. Everything is possible. And that's the principle of the membrane action as a capacitor. And now we should know the membrane potential. So the voltage between inside and outside is about minus 150 millivolt. Minus means in this case inside negative. And the membranes of bacteria and chloroplasts and mitochondria are quite comparable. They have a certain membrane capacity. That means if you have 1 square centimeter of membrane area, you will have a capacity of 1 microfarad. You don't have to know the unit here exactly. I can tell you for a bacterial cell, it would mean you need about 30,000 charges to make a membrane potential of 150 millivolts. That's it. This seems to be a large number, and the 6 protons seem to be a very small number, but let's wait for the next slide. We look at more parameters. Let's look at the transmembrane proton gradient, which is also named delta pH, and it's just nothing else but the difference between the intracellular and the pH in the outer bulk medium. And that the intracellular pHi is around 7.3 to 7.7 in many bacteria, not in all of them. And the medium might have a pH of 6.5 to 8. And that means sometimes, and it's not unusual, you don't have any delta pH. And most bacteria can perfectly live without delta pH. So that the intracellular pH is exactly like the extracellular in the bulk medium, and then there is no delta pH. And even if there is a delta pH, maybe of 1 pH unit, which would be very much and unusual, it would contribute only minus 60 millivolts to the PMF. So it's not that relevant. And if we look at the membrane potential, this is 150 millivolts. And it's built up by 30,000 charges that have to be translocated. And now comes the consideration. Just remember, we have learned the proton translocation is around 1 million protons. That means charges per second. That means the 1 million would not do very much on the delta pH, but it is enough to 30 times build up or destroy the membrane potential. And that means the membrane potential is the major player 150 to minus 60 millivolts of the PMF, and it's much more sensitive to any electrical charge that is coming in or going out. And that does not mean that the protons are not important, because they have the function as vehicles for the charges. They are the taxis that transport their charges across the membrane. Okay, let's give the answers to the questions I've raised in the beginning. What is the power that moves protons across the membrane? Okay, this is the chemical energy of metabolic reactions, mainly electron transport. That might be respiratory, aerobic, anaerobic respiration, no matter, and it might be photosynthetic electron transport as well. So it's the same principle, and this gives power to the generation of proton gradients or membrane potentials. And which gradients are building up the PMF? We have learned that. So we have the delta pH, which might be 0. It might even be plus, so that means decreasing the PMF if the delta the outer pH is like that. And the membrane potential, that is always in the negative range, and it's the larger number definitely. And why is delta pH less important than the electrical membrane potential? Okay, we have just heard it. The delta pH might be 0 or even positive, and it's hardly responding to proton translocation because it's very well buffered. And the membrane potential reacts very strongly to proton translocation. 1 million protons, as we have learned, per second would be sufficient to destroy or build up 30 times the potential of 100 millivolts per cell. So every 30 milliseconds, the whole membrane potential would be lost if the cell doesn't take care about these processes. Okay, this is my shortest summary of this presentation. Life is electric. And with that, I say thank you for watching and bye.